Konrad is a CEO of Natural Antibody company, and Konrad is also an assistant professor at SDU Denmark. And I know he has been doing some great work collecting natural antibody data sets and antibody repertoires. So I'm very much looking forward to his talk. Konrad, I'm going to shut up now and give the stage to you. Okay.
All right, fair enough. I'm going to share the screen. If you could, Sandeep, you could just confirm that you can actually see my slides. This would be
yes, I can see your slides. Yes.
Perfect. All right. Well, thank you very much, everyone, for tuning in. Thank you to the organizers for having me. And yeah, my name is Konrad Krawczyk. I'm as kindly introduced the CEO of a Natural Antibody, and I've been dealing with computational antibody methods for the last 13 years. Actually, half of that in the lab of Charlotte, who's going to talk slightly later on. So this is designed to be a very, let's say, like a generalistic talk on our efforts in the development of data and machine learning methods for therapeutic antibody development. So just some background right away. So we are not a biotech company. So there are many antibody AI biotech companies popping up right now that want to, well, essentially develop their AI platform to develop their own therapeutic program. So we are not that. We are very much tool makers. We're very much software people, methods people, and we are very much focusing on developing methods that are generalizable between different companies and different products. So we go from one company to the other, and we're building their machine learning capacity or a data science capacity, but within the antibody sphere. So essentially, I'm just putting it out there so that you get more context about where I'm coming from with some of the data and modeling obsessions that I have. So what I'm going to talk about is firstly about the data. And here I'm going to talk about our efforts into harmonizing the antibody data specifically and mapping the entire ecosystem of those. And then I'm going to talk about our efforts in the modeling. So firstly, I'm going to talk about some of the benefits and some of the caveats that we have encountered in employing machine learning models. And especially, I'm going to focus on the data aspect, of course. And one thing that I'm actually going to keep talking about, because I think that this is actually important at this stage, is the difference between some of the methods and data sets being the facilitator of the antibody discovery process versus actually all of those methods and all of our effort into modeling and our I'm not saying only myself, but in the entire community, into actually having the computational methods as broadly defined as you want, being the driver of the discovery process. So for our efforts, we have divided everything into the three pillars. Firstly, the data, and we start with a very, let's say, ambitious premise, get all the data ourselves, which, it's something that you need to do if you want to perform any kind of modeling. Yeah, and this was supposed to get, I'm not going to abuse the word literally, but as close to getting all the data as possible that is out there from all the major repositories, harmonize it, put it in one place, and then employ it for modeling. Then, of course, models. There are many groups out there that are performing absolutely amazing work, but we are not in the business of, just picking up someone's GitHub repository and running with it. We actually want to know what is happening, because if we are developing a method somewhere within on a partner platform, we do need to know why the model fails and whether we need to adjust the model for their specific use cases. And then finally, data and models are all great, but quite a huge majority of everything that's happening in pharma companies these days is still very much rooted in experimental methods. So the data and the models, if we are using them, it's great. But if we want to put them in hands of people actually at those companies that are using them, then they need to be made in a very, very hassle-free way. And I'm not only talking about nice GUIs, but actually so that they do not need to double guess the parameters. Yeah, I'm not an experimental person. If someone just puts me on a bench with a protocol, I'm not going to perform well. Yeah, it works the same way with data and machine learning. So just putting some more concrete terms on all of these. With respect to the data, like we mapped all the major repositories that have the different antibody data sets. And of course, like we are complementing them with harmonization methods like our knowledge graph, and also generating our own experimental data together with our lab-based partners. Within the models, we are constantly making efforts into developing new methods that tackle specifically therapeutic antibody development, but also staying on top of what is currently new and could improve the process, as well as improving the current methods as all the data sets are coming in. And of course, within the platform, we are distinguishing between the mundane antibody search engine, because we have all the data, then it's easy to just make them searchable. And then difference between the single molecule views. So if you want to just engineer a single molecule versus filtering multiple. For the purpose of this talk, I'm mostly going to talk about those two stages rather than this one. So I'm going to kick off with the data. So as you can imagine, and this is the question that is being posed to us quite often, do we, for instance, get, I don't know, aggregation or stability data that we mine from the literature? In some cases, we might, but harmonizing the data from major sources actually does not entail that, because you need to make sure what is the common denominator between all of those. And even if you pick several publications that have even data from the same assay, and they might not be that comparable, if you know what I mean. So what we focus very much on is what are the systematization efforts, and how to make all of this maintainable. Yeah, because all of those repositories, especially the public ones, they are constantly changing, and you are ending up with very much in a whack-a-mole situation. I'm going to give an example with the structural data on that. So our efforts here, we mapped the repositories into the ones presented here. So of course there are the major repositories like GenBank, structure. So this means the PDB, NGS. These are the SRA repositories, and some of them are more amenable to automated retrievals than not. Structures and GenBank are actually not that difficult. Patents, this was a lot of work. Mining data from literature, even though NLP methods are really making progress, it's still far from perfect, and we actually have people doing the manual annotation. Similar with therapeutics. For NGS data, we started up with a combination of automated and manual. Right now, we're moving very, very much towards the automated annotation. And for all of those, we took the common denominator as the V region sequence. Yeah, and of course, in some cases, people just post just the CDRH3 sequences or portions of these sequences. We had to put the cutoff somewhere. Also, when you actually get all of those data sets, it's we are not stopping there. We are dividing the data that we get, but we are also augmenting them with new information. So for searches, we have different indexes and then embedding. So Lucy gave a really great introduction to encoding protein sequences. This is what we're doing for antibodies. And what we are also doing is for the text information, because this is everything dealing with antibody sequences, we are adding certain information to the text that we're getting. So for instance, for patents or from the literature where we get paper titles or abstracts, we are adding, for instance, name entity recognition layer, so that you can see what molecules were identified there, but also the synonymization layer, so that different names of proteins, genes can be put in one equivalence class. And just to give you some numbers, currently, we have more than 800 therapeutics, close to 300,000 patent sequences. more than 6,500 structural antibodies, more than 170,000 sequences from GenBank. For NGS, around like 250 bioprojects. Like this adds up to like 25 billion row reads. I'm going to get back to that like when I'm talking about the embeddings. And from the literature, we have around like 7,000 sequences that are attached with diverse targets. And of course, we also generate some data with our experimental collaborators. And sometimes, some of those just percolate into the public space. So if you're interested in some of these, I do recommend if you look at the papers on the left here. So it's all great, having all of this in one place, but it's not that you have a pipeline, you have just one button and it's going to keep merrily updating all of your data. So you need to keep an eye on it, and this is the unfortunate truth about that. So I'm pretty sure that Charlotte later on is going to mention SABDAB. So SABDAB was released around 2013. This is where James was coding it up, and as the structural data was, obviously, was growing, but then, the nature of the data set was changing together with it. Obviously, there are mundane things outdated entries. There are some weird chain names that even Roland Dunbrack was complaining about on Twitter. And then you have some even more you see things that are quite specific to the modeling tasks that can be down the line. So the two bars that you can see here is when I'm clustering the available antibodies by a unique antigen, or whether I'm allowing multiple antibodies per antigen. So obviously, if there are quite a lot of unique antigens within the structural space, then those two bars should be similar. But then as we're progressing within the years, this difference became much smaller. And of course, this is not only due to the fact that right now we have quite a lot of coronavirus antibodies against the same target, this trend started much earlier. But if you want to squeeze something out of this data set for structural predictions or binding predictions, then this is the kind of thing that you do need to monitor. So obviously, before we go to the modeling, quite an obvious aspect of this is building a search engine, and this is what we have built, and we made it very much antibody specific, so that it is not a blast search, it's a specific antibody specific index which we're using and our partners are using for looking for V regions, DRH3 sequences, but also for text-based retrievals. So we are generally just using it as a single point of entry towards data in the public domain. And I'm never going to claim that we have all of the data, but we are making efforts to at least have the coverage that is as close as possible. And in the spectrum of facilitator or the driver of discovery, this is a typical facilitator, yeah. But I still think that using a tool like that, for instance, for us, it allows us to help people at least making decisions that are very much informed with respect to what is out there in the public domain. Then finally, we can move to the modeling part. So now that I have the data, I can start modeling. And of course, for the models, there are the considerations of what is the question that the model should be answering. Yeah, and this is slightly different between whether I'm answering an academic task that I want to develop, a structure predictor or a developability filter, or whether I'm developing a model that is actually very specific to a given therapeutic case. Yeah, because for instance, the partner that I'm dealing with, might not be very interested in kappa chains, but is very, very interested in lambda chains. So picking the right model and the right data are quite obvious, but also one thing that I was slightly surprised about is that there is a shift in the frameworks used, and when we're doing a review of the deep learning methods and antibody specifically recently, we could see that it's a, I thought that just by my hunch was that the TensorFlow is going to be used much more, but at least according to our research and antibodies, in PyTorch is much more used. We're essentially using both. So here again, what we are covering is a very broad spectrum of models, and we are dealing with, let's say, the older methods that we want to improve. There are some new methods that we want to tackle, and then there's the entire new frontier that we would like to explore. So on all of those three, I'm going to talk first about, let's say, old methods. Maybe they're maybe not old methods, but old problems. You should phrase it like that. So here I'm going to talk about structural modeling and developability prediction. Some new concepts, so positional predictions. Perhaps it's not that new a concept, but in dealing with this and machine learning terms, I think that this is somewhat not novel. And then, of course, the next frontier, according to me, this is very much in the down the alley that Lucy was talking about. So actually embedding the entire antibody space and learning from this latent space, what are the features of antibodies, how you can use this information to improve the current models, but also notwithstanding generating de novo molecules. So first, let's just go to the all methods but fast. So here we're talking about structural modeling, and this was until, let's say, somewhat recently, in the domain of homology modeling, and the methods were very nice, and they were actually back in the day, when we were talking about tens of seconds per model, and then we were considering it fast. Yeah. So what some of the deep learning methods that are tackling this problem are doing, and I suggest that you can look at this publication again, because we are reviewing that, is they can generate the coordinates, or they can generate the constraints for the coordinates, distance matrices or dihedral angles, but sometimes they just get the physics quite wrong. Yeah, so you can actually produce an okay backbone, okay with respect to the objective task of the template that you were supposed to model, but sometimes the atoms are just not going to be either they're going to be too far away or too close, and it's generally not going to be very physical. So current solutions to that are that you generally spit out your models, your coordinates or constraints quite fast, and then you generate the model that is let's say very physically kosher, and you can use energy-based functions for that. However, we took a slightly different approach. So we firstly thought of training the model to actually understand what it means that you should not put different atoms too close or too far away from each other, and just then we were focusing the predictions on making sure that you're actually getting the sequence coordinate mapping right. And of course, this is not a panacea. It's, I must say that this is producing the predictions fast, and it's producing the predictions that are physically plausible. But of course, in some outlier cases, this producing coordinates that are quite not physical, but they're pretty easy to pick out. And of course, this is something that we are struggling with. So a Rosetta dataset is something that quite a lot of people are dealing with. If we're talking about the modeling of H3, we could get something in 2.1 angstrom on that, which is very much down the ballpark of all the methods that are coming out right now. However, if we're looking at some of the hard cases that are coming out, especially in very much longer loops, or especially in some of the new nanobodies that have very long loops, then sometimes, of course, we fail miserably. Yeah, so this is just to say that in the case of structural modeling, there is definitely a way of making the predictions fast by training the model into how the physical connections should look first, and then just focusing the model on the objective task of predicting the right coordinates. Then, of course, the following the punch means developability, and some of the methods that were developed back in the day, like one very good one, so TAP by a former a member of a lab I was in, Matt Raybould, and a developability index, they were very good, but they looked at especially the TAP method, looked at the structure to make the calls for some of the developability metrics. And this is all great, but then you do have the stopping stage of needing the structural model. And it's all good if you get to the point where you have very fast structural modeling, but in some cases you might even just want to just let's say leapfrog even that, but it's not that easy. So you cannot simply just pick your even a small convolutional network or a dense network and just make sure that you reproduce some of the scores. Yeah, so you need to use the models that are quite reminiscent of the objective test that you're supposed to model. For instance, the modeling of CDR-H3. Otherwise, it's not simply does not generalize quite well. So in those terms, having this, we can produce quite fast developability calls for different metrics. And right now, we can do it, for instance, for 200 molecules per second. So this is something that we were benchmarking on our laptop. So just full disclosure, we have pretty good laptops, but right now, we can actually do 1 million sequences in pretty much 1.5 hours. And this right now becomes quite achievable for the high throughput screening, because back in the day we had to struggle quite a bit with different infrastructures and how to play with that so that the methods are up to speed. And of course so the main conclusion from this is that if you create if you make those methods to run much faster but still not sacrificing the precision, then you can fulfill the task of de-risking your molecules at quite an early stage. But again, this is I see this as a huge facilitator, but still not the driver of the entire process. So perhaps right now, we're going to proceed to the new concept, or at least somewhat new, the positional predictions. So as you know, there are many NGS data sets coming out. So we are getting a very, very good coverage of what is the natural space that is available to antibodies. And of course, together with some older methods, there are some predictions out there that allow you to make calls for, say, developability, immunogenicity, whether something looks very much human, whether it doesn't, and it doesn't correlate with the ADA scores that you can expect from your molecule. So of course, one thing that I would like to do is when I have a sequence is to modify it in a way that it reduces the some objective score, such as say immunogenicity. And of course, I can do it in let's say I can enumerate everything, all the positions and all the amino acids there, and see whether it makes sense with regard to the score. But I'm always going to get some score, yeah. But it does not mean that any kind of enumeration of residues in CDR-H3 or any other CDR, even not even talking about the framework, is actually going to be biologically plausible. So for this reason, we are started making predictions for actually what are your mutational choices. So this was one of the first tools that we released, and it was very much, it is not a deep learning tool, and this is very much a statistical breakdown of the sequences in the observed antibody space that tells you in this position, you're looking at several 100,000 sequences, you have those mutational choices. Yeah. What it doesn't tell you, because of the statistics, is what is the interplay between the different positions. Yeah. this is the kind of information that is picked up by like a quite hefty BERT models like anti-Berta, anti-Berti, ABLANC, which are masculine language modeling based. And I do suggest that like you guys look at that, but like you know, most of those models like were actually like not designed for predicting the different positions, or at least like you know, those that were masked, but essentially like, you know, using this kind of problem to perform to get the encoding of the sequences, and then to model the latent space of the antibodies. Yeah. So one thing that we were exploring is like, you know, whether, you know, if you actually like do not want to model the latent space, like, can you actually get away and making the predictions in let's say like, you know, with the smaller models. And why do we want a smaller model? Well, because you know, computations cost, and like, you know, the smaller model we have, then the faster it's usually going to run for. So for instance, like if we are looking at objective task of just like, you know, looking at the positional predictions, then our smaller models, you know, surprisingly, like, and you know, I don't like this fact, are not even that much greater than just, you know, the statistical based model of that we developed before. But then like ABLANC that like we were using like quite a lot, it's actually like a great tool. I suggest that you guys have a look. It has 85 million parameters, but for some of the tasks, like, you know, the overall positional prediction, it does better than the smaller networks, but not that much. So you can see that it essentially like it performs better in CDR H3s, but the difference in the performance is actually like was quite surprising to me, like, given the numbers of parameters. And also, like, you know, just, you know, saying that, like, you know, the bigger BERT models, like, they're designed to actually you know, like, you know, for much more complicated tasks than just, you know, dispositional predictions. Yeah, so like, you know, here we're just using like what are the mutational choices, and like, you know, how small a network can we get away with. So of course, like, what this allows you to do is, if you're doing humanization, then going beyond the germlines. If you are doing something that, you know, say like, you know, you want to really remove liabilities from your sequence, then you see like what is the biologically plausible space. And of course, like, you know, going beyond simple statistics like AppDiver, the deep learning methods, they allow you to actually map the dependencies within the positions. Yeah, so like in which position affects which one, and make positional prediction calls based on this. But again, like, you know, this is not really the driver, but it's the facilitator of research decisions. So finally, the next frontier, and this is something like, you know, this is my opinion, but, you know, I hope that like I'm not the only one within this, is learning the antibody latent space. So using huge pre-trained models like, you know, anti-Berta, ABLANC, and so on, you can actually use this as a basis for improving quite a lot of tasks. Yeah, so like, and there were examples recently from Pareto prediction, from structural predictions, and from affinity-based predictions. However, for training those models, you need a lot of natural sequence data. Yeah, what we tested was that, like, you know, if you're using like, you know, some of the, you know, even smaller models or the bigger ones, whether you still perform well on other types of data sets that are not natural, like patents, and you do not. Yeah, so this is something that, you know, I still think that like it needs to be taken into account. Perhaps like, you know, fine tuning is going to deal with this, but it seems that like in natural data sets and like in therapeutic data sets, like, you know, even though they have some commonalities, like, and there might be like, you know, just, you know, domain differences that need to be surmounted. Finally, again, like, you know, small model, like, you know, it's really important, like in what, you know, what you're asking for. Yeah, because, you know, we are dealing quite a lot with embeddings, and like we want to see like, you know, what are the limits of you know, different ablations, like, you know, if you're looking at the number of sequences you're putting into this, or the number of parameters you're dealing with it. So for tasks like you know, just like in looking at whether I can train a model and reproduce some like, you know, cool pictures, like whether like, you know, the genes are separated on my t-SNE plot. Yeah, even like in a very small network that has encodes the antibody in 128 dimensions, like, it can do it. But of course, like, it's going to fail absolutely miserably if you want to do any transfer learning with it. Yeah. And of course, like, this is what leads me to the conclusion of that. It's like, what we are doing quite a lot with is training those embeddings on our NGS data sets. But again, you need to remember, like, what we were dealing with. And even though from the last 2 years, we have 138 bioprojects, which is like, almost 3 times as much as was like when we were developing the observed antibody space. Within those bioprojects, as you can imagine, like, there's a lot of coronavirus data. Yeah. So again, like, we need to think quite carefully, not only about the difference between the natural data sets and its effect on the therapeutics and whether those are 2 different domains, but also like on the different biases within the natural data itself. All right, and I think that, like, this is essentially the possible driver of the discovery process, because this will allow you to encode the desired properties, like sequences that are more developable. You'll be able to model the space so that you can actually literally, like, sample novel sequences, and of course improve the low N learning. So just concluding everything, I hope that, like, you get the following messages home, that the data that is out there, like, it's not beyond reach. Yeah, like, especially, like, it took us quite a lot of effort, but like, we did manage to harmonize it. So at least, like, having all this data, like, it allows us not to fly in the dark in any discovery process. And the current models, like, we can actually, like, make them, like, fast enough to actually make the filtering of high throughput data sets. And of course, like, I do firmly believe that the next frontier is very much in learning the Latin space, but then, like, ending on the, like, in a slightly more let's say, like, cultural note. I do think that like, this allows, like, quite a cultural change, and, like, going from like the experimental first to, like, very much embracing the computational paradigms. And yeah, if you guys, like, are interested in anything that I said in, like, a more technical detail, or, like, would like to work with us, like, just do give me a shout on this email. Thank you.
Thank you so much, Konrad. This was really a great talk. I think there is one question in the chat for you. Is from Christian Henrich. Now that all antigen structures are predicted and only HCDR3 predictions remain a challenge, how long until epitope prediction is reliable and easy? Would not antigen structure inform CDR3 prediction? Would one do physical docking of predicted antibody and antigen, or try to predict complex antigen-antibody like Baker did for protein-protein complexes? Thanks.
Yeah, so I'm going to take it apart. Like, the first part, CDRH3, I'm having the philosophical debates whether you can actually, like, how much better than 2 angstroms can you get? Yeah, so that's everything I'm going to say. Yeah, because I did put the disclaimer at the end that sometimes, like, when there is a new structure of CDRH3, we just need to fail on it. Yeah, and it might just be because it's slightly more flexible. With respect to epitope prediction, I must say that, like, I do have huge hopes for the AlphaFold, because essentially, like, what it did, like, if it really, like, did, like, what I think, it's actually, like, mapped all the available, it extended significantly, like, our knowledge of the possible, let's say, like, a druggable, antibody druggable surfaces. Yeah. So I think that, like, using this information is definitely going to help with epitope prediction. At what point we're going to have epitope predictions that are, as close as experimental ones? Sorry, I really do not want to play a futurist here.
Okay, thank you, Konrad. I do have one question of my own for you, if I can ask. It's about NGS repertoires.
Yeah.
Typically, we lack the VHVL information, pairing information over there. Are there attempts made by data annotators and data people who produce this data to kind of determine the VHVL pairings also along
Yes.
with the NGS data?
Yes, so this is already like in Charlotte's lab, an OAS database. Like, you guys can go and like, and there are many paired NGS data sets over there. It's pretty good. And yeah, like, and we're also we found internally around like 30 of those. Yeah, that like, and we were processing it ourselves.
Okay.
But yeah, but essentially like, and there are more and more coming.
Okay, thank you. So this actually brings us to the end of the first session, and we'll now take a break.